All right, well, we're about 1 minute past and we have over 20 participants so far, so I will get us started. So welcome. So for anyone that is attending this call for the first time, we hold this industry-wide call every month for any company that really wants to participate in the cultivated meat value chain. And the purpose of these calls is really to introduce and welcome new solutions providers into the industry with the ultimate end goal of establishing new collaborations that are going to accelerate your R&D as well as commercialization. So if your group is interested in presenting on a future call, or perhaps you're working with a company that could benefit from being on these calls, please do feel free to put them in touch with me directly and we can get them added. So just a few housekeeping items from our end today. I wanted to share that we released our 2022 State of the Industry reports earlier this month. So these reports we publish annually, and they cover really the latest on industry advancement, investment, R&D progress, policy, and regulations. They're free to download, so I'll drop a link in the chat in case you haven't seen them or you'd like to share them. They can be accessed there. Also, we sort of base a lot of the information in these reports off of our publicly accessible company database. And so please don't forget to add yourself to this database to ensure that our data and record keeping is as accurate as possible. And then I also just wanted to share, there's a few links in case you're interested to recent webinars that we've hosted. One is on the environmental impact of cultivated meat based off of a study that we published earlier in January, which can be accessed at this link here. And then also a link to the webinar for the State of the Industry report that we held last week. So hopefully those resources can be useful to you in some way. And I'll get started on introducing today's speakers. So I'm pleased to welcome Kosuke Minamihata and Jes Kuruvilla from PeptiGrowth, who are going to be speaking today about their peptide alternatives to growth factors. And then we'll turn it over to Steve Reese from Defined Bioscience, who will be sharing more about their growth media and inputs for cultivated meat. So please do feel free to drop questions in the chat throughout, and we'll be able to get to them at the end of each presentation. For this first presentation, because it's super late in Japan, we have it pre-recorded, but both of Jes and Kosuke are here to answer questions that you might have at the end. So Renee, if you wanted to share the presentation.
Thank you everyone for joining our webinar today. My name is Kosuke Minamihata, and I work as a technical support manager for marketing at PeptiGrowth. In this webinar, I and Jes will be explaining about peptides that can substitute growth factors, which are very important in the regenerative medicine, cell therapy, and also cultivated meat industries. So let's get started with the explanation. First, let me give you an overview of our company. We are PeptiGrowth, and we are established in April 2020 through a co-founding partnership between the Mitsubishi Corporation and PeptiDream. And this PeptiDream is a biopharmaceutical company and that specializes in peptide-based APIs and possesses a proprietary technology called PDPS, and which stands for Peptide Discovery Platform System. And this PDPS technology allows us to screen out the peptide, cyclic peptides that can strongly and specifically bind to the target molecules, and those are typically receptor proteins or growth factors. And unlike other peptide screening systems, this PDPS system and PDPS technology can handle many kinds of non-natural amino acids, and resulting in a much larger diversity of peptide libraries. And therefore, the hit rate of this PDPS is much higher than the conventional peptide screening systems. So at PeptiGrowth, we utilize this PDPS technology to create peptide that works as the same function as the conventional growth factors and cytokines. And our mission is to bring innovations to the cell therapy and regenerative medicine and cultured meat industries by utilizing those functional peptides. Here's the conceptual diagram of what we do. Growth factors required for cell culturings are generally very unstable, and there's an issue of lot-to-lot activity variations. And this is particularly problematic in clinical studies and that requires GMP controls. And furthermore, some growth factors are produced in biotic conditions and may contain animal components, making them unsuitable for clinical use or as additives in cultured meat. The scalability of most of the growth factors are not really high, so resulting in extremely high prices. And we are solving, these problems associated with conventional growth factors by utilizing functional cyclic peptides. And our peptides are in the form of cyclic peptides with some non-natural amino acid in it, so making them very, very highly stable against the degradation of proteases. And also, and since they are just cyclic peptide without any tertiary structures, they are highly stable even at high temperature conditions and do not denature like conventional growth factors do. They are completely chemically synthesized, so we can eliminate the issue of lot-to-lot activity variation, and they are all animal component free. Also, since we already have very large facilities for making peptide, the production scale can be easily increased. And by using such large facilities, we can significantly reduce the production cost of our peptide, and we can provide them at a reasonable price. So in particular, the cultivated meat industry requires large quantities of growth factors at low price. And we believe that our peptides are the only solution that can meet such difficult demands from the cultivated meat industry. So we will support your cell proliferation and cell differentiation process to make your final product by using a cyclic peptide. So we have currently launched 6 products named PG001 to PG006. And these products include an HGF alternative peptide, TGF-beta1 inhibitor peptide, BDNF alternative peptide, a Nogi-like peptide, and also BMP7 selective inhibitor and BMP4 selective inhibitors. And our most recent product is PG005 and PG006 are very unique peptide that we launched in December 2022. And we are continually developing various types of growth factors, alternative peptide, with the goal of launching 3 to 4 products per year. So let me explain how our peptide work as agonist or antagonist by introducing our peptide one by one. First of all, I'd like to explain about the PG001 HGF alternative peptide, which is a c-Met agonist. So this is an example of agonistic peptide, and in nature, this HGF hepatocyte growth factor binds to its receptor called c-Met and induces the dimerization of c-Met receptors. And this dimerization process is the trigger for the proliferation and following subsequent signaling event in the cells. And our PG001 does the same thing as HGF does. Upon binding to the c-Met receptors, PG001 induces the dimerization of c-Met. And our peptides are not mimic of conventional growth factors or the minimized form of growth factors. Instead, we screen peptide that can specifically bind to the target molecules. And in the case of this PG001, the target molecule was c-Met. And we screened cyclic peptide and then connected them using the appropriate lengths of linkers so that those peptides can induce the dimerization of receptors. So what I want to say here is that sequences of peptides are entirely new and original. The graph below shows basic experimental data of c-Met phosphorylation and also the Hubeck proliferation, comparing the HGF and PG001 activities. As you can see, as our PG001 showed comparable activity to conventional HGF in terms of molarity. Usually, the x-axis of such dose-dependent assay is in the unit of nanogram per milliliter. But since our peptides are relatively small compared to the conventional growth factors, and this, in case of this PG01, its molecular weight is approximately 1/16 of the HGF. So, for example, this 2 microgram of our peptides is actually equivalent to 32 microgram of HGF. So you may think the price of a peptide is not reasonable by looking at its quantity, but if you compare the price of the equivalent amount of the corresponding growth factors, I believe the price of a peptide is competitive. Here we show further activity evaluation data for PG01, which were obtained by Professor Taniguchi at the University of Tokyo. And he's an expert in regenerative medicine for liver regeneration using the human iPSCs. And he conducted the proliferation assay and also the cloning assay of the iPSC-derived liver cells using either conventional HGF or our peptide PG01. And the results showed that the activity of PG01 and HGF are comparable to each other. And moreover, in the result of this cloning assay, the number of colonies found using PG01 after 10 days of incubation was statistically higher than the case of using HGF. And in fact, this Professor Taniguchi mentioned that he has faced lot-to-lot activity variation issues with conventional growth factors, and he decided to replace it with PG01. The next product is PG02, which is a TGF-beta1 inhibitor peptide. This is an example of an antagonistic peptide. And TGF-beta signaling regulates cell fate, such as cell survival, proliferation, and differentiation. And so TGF-beta1 inhibition is often used in cell differentiation processes. The general approach is to use small kinase inhibitors such as AAD301 and SP52534. And those small molecules enter cells and interact with receptors to inhibit phosphorylations. However, since they're small molecules and lack specificity, they affect other kinds of receptors as well. So this low specificity of small kinase inhibitors sometimes causes problems such as cytotoxicity. And another approach is to use neutralizing antibodies that works outside of cells, and antibodies specifically block activation of TGF-beta1 signal. However, these antibodies are usually made by mammalian cells such as CHO cells, so they are not animal component free, and their price is extremely expensive. So our PG02 can solve these problems associated with small kinase inhibitors and antibodies. And PG02 has no membrane permeability, so they work outside of the cells and interact with TGF-beta1 specifically. And it is chemically synthesized and therefore animal component free, and we can provide it at reasonable price. And this SBE reporter assay showed that our PG02 exhibited comparable inhibitory activity to the antibody. And this PG02 can be used as an alternative for small kinase inhibitors. The third product, PG03, which is a BDNF alternative peptide. And this is another example of an agonistic peptide, and its working mechanism is the same as that of PG01. The target molecule is this TrkB receptor, and PG03 induces the dimerization of TrkB receptors. The left-down figures shows the result of TrkB phosphorylation and also the reporter gene activation assay, and comparing the activities of BDNF and PG03. And as you can see, the activity of the BDNF and our PG03 are comparable each other. And in this reported gene activation assay, our PG03 showed even higher activity than BDNF. And due to the molecular weight differences, the 10 microgram of PG03 is equivalent to 25 microgram of BDNF. This in PG03, we have some application data obtained by third party. And this is the data obtained by Zigzag Bioengineering by using their proprietary technology called nerve organoid. And they are developing the drug screening platform or regenerative medicine for nerve injuries. And Zigzag Bioengineering evaluated the length and the width of neural axon bundles generated from the cellular aggregates by the effect of PG03 or BDNF. And our PG03 induced the generation of the neural axon bundle comparable to the BDNF, and the quantitative results showed that our PG03 actually generated a longer and thicker nerve axon bundles than BDNF. And here's another application data of PG03. We evaluated the behavior of neurite outgrowths from the SHSY5 cells induced by BDNF or PG03. As the dose of BDNF or PG03 increases, the cells started forming dendritic structures, and the quantification analysis of the images shows that PG03 and BDNF are comparable to each other. And these data support that our PG03 possesses equivalent activity not only in the basic signaling activation, but also in higher order phenotypes of neural cells. The next one, PG04, noggin-like peptide. And noggin is a protein that interact with various kinds of BMP family proteins such as BMP4 and BMP7 and inhibit their signaling. And noggin is often used in development of organoids such as lung, intestine, and kidney. Our PG204 also work as similar to noggin and interacting with BMP4 and BMP7 families and exerts inhibitory activity. The left down figures shows the result of inhibition experiment against BMP4 and BMP7 using either noggin or PG204. And as you can see, as our PG04 showed the same level of inhibitory activity as noggin in molar basis. And since the molecular weight of PG04 is actually 1/8 of the noggin, so this 5 microgram of PG04 is equivalent to 40 microgram of recombinant noggin. And closely related to the previous mentioned PG04, we have recently launched 2 new products called PG05 and PG06, which are BMP7 selective inhibitor and BMP4 selective inhibitor peptide, respectively. And these are entirely new functional peptide, and PG05 and 6 allow us to selectively control the signaling of BMP4 and BMP7 families. And the selective control of BMP signaling was not possible with the conventional noggin or small kinase inhibitors. So those PG05 and 6 are very unique functional peptide. Although we are not clear about how to utilize these 2 product peptides, but we anticipate that they can be useful for detailed analysis and regulation of BMP signaling, as well as for the development of novel cell differentiation processes. So these are the result of the PG05 and 6. PG05 is a BMP7 selective inhibitor, so it showed inhibitory activity comparable to the noggin against BMP7, but it showed non-inhibitory activity against BMP4, even at the 10 times or more higher concentration than the biologically active concentration of noggin. And on the other hand, PG06 is a BMP4 selective inhibitor, and it inhibited the BMP4 at the same level as noggin, but it showed no inhibitory activity against BMP7. And these are our product pipelines. As I mentioned earlier, we have launched 6 products so far, PG01 to PG06. And as upcoming growth factors, which we are expecting to launch in this year 2023 or the early of 2024, we have several major growth factors like EGF, basic EGF, and VEGF, and Wnt3A. We are also developing many other, peptides that those are listed here, which includes like TGF-beta superfamilies such as activin A and BMP4 and TGF-beta1, and also interleukin cities like IL-2 as well. So if there is any growth factors either in or outside of our list, which you have great need, we would be happy to co-develop them with you. So please let us know if you have interested in such a co-development program. Lastly, I would like to explain our peptide supply system. And PG peptides are manufactured by our sister company called PeptiStar, located in City, Osaka, Japan. And PeptiStar is a CDMO company for peptide APIs and originally established to make the peptide APIs developed by our parent company, PeptiDream. As PeptiStar has the GMP compliant facility for API production, our PG peptides can also be made in GMP manner upon demand. So furthermore, PeptiStar has large manufacturing and purification facilities for peptide with a maximum production scale of 10 kilograms per batch. So by using such huge facilities, we can supply a huge quantity of PG peptides at reasonable price. Here's a summary of today's webinar, showing the benefits of using our PG peptides. Firstly, PG peptides offer an outstanding cost advantage over conventional growth factors. PG peptides have high stability, which means that media exchange can be reduced. This is a significant advantage as it reduces the need for frequent replacement and can save time and resources. PG peptides are also animal component free, which is an ideal feature for the use in regenerative medicine, cell therapy, and cultivated meat industries. Although, in my understanding, the growth factors used in production of cultivated meat do not need to be GMP grade, we can provide our PG peptides under GMP control upon request, ensuring high quality production standards, and also we ensure the timely delivery. And we also offer co-development of alternative peptides to any growth factors and cytokines. Our products have started selling on scientist.com since April 2023, and we would like to take this opportunity to encourage you to visit the scientist.com and consider purchasing our products. Our PG001 to PG004 are currently available for purchase. If you are interested in PG005 and PG006, please note that they are not yet available on scientist.com. However, please feel free to contact us for any inquiries about these products. And additionally, for bulk orders, please contact us directly as we can provide you with the price indications. And here's our contact information. Please feel free to contact us if you have any inquiries. Jason and I are happy to answer any questions you may have during this webinar, and thank you very much for listening.
That was very clear. Thank you for that webinar pre-recording. Well, Jes and Kosuke, thank you so much for being here and answering questions. People can follow up with you and get in touch with the contact information on the slides. So thanks again for sharing some of that information today. And we can, I think, pass it over to Steve.
Sounds good. Okay. Good morning and afternoon and very late evening, especially for the PeptiGrowth folks. Thanks for tuning in today. I wanted to give an overview of what Defined has currently been up to, especially in the cultivated meat space. So these will mostly be highlights of a few things that we've been working on and what our products are currently doing in the space, and some exciting recent developments as well that I'm happy to share. So if there's any questions as well, I won't go too heavily on the technical side for too much of this, but we're happy to do that in the Q&A and with follow-ups after this call. And I think Jerome from our side is here as well. He'll be more useful for some of the cellular biology questions than I will. So for those that don't know, Defined is a company that started in 2019 in San Diego, California. We're a team of 8 folks here, and our goal is to be a provider of growth media ingredients for science, therapeutics, and the food industry as well, especially cultivated meat. Currently, we have medium formulations and growth factors focusing on pluripotency at the moment, and that's expanding this year into a larger catalog of media and growth factors exploring pluripotency and differentiation and other parts of that process, as well as offering custom services for protein production, medium optimization, and medium formulation as well. And that's for research grade as well as CGMP production. So if you guys ever have questions on those and you don't see it on our website, don't hesitate to reach out. Our focus specifically is on products that are defined and animal origin free for culture of cells. And we've started with stem cell products as kind of a beachhead. Our goal in providing defined and animal origin free solutions are that we wanted to offer products that are consistent and reproducible. So when you're introducing things like animal serum or undefined componentry, sometimes you don't know what you're adding to your cells at any given time. And you can see a lot of variability throughout that year as you change lots on those products. And these are things that we've known for a long time in the stem cell industry for life sciences and pharma. And it's something that Cultivated Meat has been tackling from the beginning, trying to get into these formulations as soon as possible. So it's been encouraging to see that kind of transition early on for that space. So to highlight two of the products that we have in our catalog, one of those is high-def B8, which is something I think a lot of folks on this call are familiar with. It's a low-cost stem cell maintenance medium that was developed by the Burridge Lab at Northwestern University, which they published in 2020, I believe. And this is something that Defined currently offers and sells. It's a weekend-free medium, so it uses a thermostable version of FGF2, which we've been seeing enables longer stability of that medium at 37 degrees. So you can do 2 passages and 2 medium changes the day after within a week and use enough medium for those off days so that you're not going in on the weekends to change your medium. It's fully defined, the formulation that we make, so it's fully animal and serum free. There's no hidden components. You can see the recipe for this right on our website, and speaking with Paul as well and looking at his publications using this medium. And we offer it as a 400x supplement. So rather than sending you a full half-liter bottle, we provide this as just a cryo vial that you would thaw and use directly on site, adding to your own DMEMF12. So the table on the right is comparing B8 to the E8 medium that was used as the scaffold for developing B8. So both media are animal albumin-free and use defined ingredients, but B8 has that advantage in that weekend-free context. It's also been validated for higher passages. So at the time that Paul had published the original work on this, he had taken a cell line out to over 130 passages, and the cells looked just as good as they did at early passage counts. At the time, I think E8 had only been validated in the literature up to about 50. So it's not to say that E8 couldn't reach those yields, but we had seen that validation pretty strongly for B8. Additionally, FGF2G3 is the thermostable form of FGF2 that we offer. It's sold separately as well as included in the high-def B8 formulation. So this protein takes the original sequence of native human FGF2 and introduces 9 point mutations. This is out of work from Masaryk University in the Czech Republic back in 2018. And these mutations were found to enhance the function and performance of FGF2 wild type, so that this protein, the wild type form, typically has a half-life of about 10 to 12 hours at 37 degrees Celsius, which is the temperature where we're really talking about for mammalian culture. But these mutations improve that half-life from 10 to 12 hours to over 3 weeks at the same temperature. So this is one of the biggest drivers for likely enabling this weekend-free capacity of the B8 medium. So we offer this right now on the site individually for sale. It's in microgram and milligram quantities, but for grams and higher, we offer this as well. Just don't hesitate to reach out. I wanted to highlight a few times that some of our products have been in the literature as well, particularly recently. So Andrew Stout and David Kaplan's lab at Tufts published on a Beefy 9 medium, which took the B8 medium as a scaffold for bovine satellite cell culture, and found that after testing several additives, adding recombinant albumin was able to improve performance of that medium. This paper was an excellent effort looking at a lot of different variants that could be added to this formulation to improve cell growth. Currently, the Kaplan lab is looking at some of these alternatives for lower cost sourcing of albumin or albumin-like components to try to improve sustainability. Obviously, recombinant albumin is a pricey alternative compared to other sources that could do this same function. And animal-sourced albumin is certainly an option, but this kind of defeats the point when we're talking about getting away from animal-derived componentry for the cultivated meat space. So these alternatives are certainly critical. Additionally, from the Kaplan lab, this was recently accepted is the BFR medium. I think the bioarchive is also still available, but this was kind of expanding on that same story from the Kaplan lab. So instead of using recombinant albumin, what else can we source that could potentially perform in a similar way? And they tried using plant isolates, so plant protein isolates from a variety of agriculturally relevant crops, and found that rapeseed protein isolate in particular performed very well from a mg per mil additive perspective. So they screened a wide range of crops, and rapeseed protein isolate wasn't necessarily the cheapest form per kilogram of crop and not the most protein yield present per amount of extract, but it had the best performance when tested on a mg per mil perspective. So this is something that the team is still exploring, as well as looking into other additives that have been suggested in the space previously. Additionally, there is a paper that's in bioarchive as well that's still being peer reviewed from ACIB. So that's looking at Alex and the Harold Pickler Labs group, looking at other additives to B8 and other formulations for bovine culture as well, as another way to try to replace some of these more costly components. So it's excited to see B8 being used in these contexts and looking at the next generation of what this medium could look like in the context of cultivated meat production. I also wanted to highlight some work on the human IPS side, which could be relevant for when we consider characterizing cells that are grown in B8 or similar formulations. So obviously cell sorting and flow cytometry are going to be critical for characterizing the cells that are derived from cultivated meat sources, as well as the recovery of those cells afterwards. There's a variety of harsh and softer methods of sorting, and we worked with a company in San Diego called NanoSelect Biomedical, which produces the Wolf G2 sorter, which is a gentler method of cell sorting in the context of sorting stem cells that were grown in high-def B8 medium. If you're more interested in this work, we have an app note of our work with NanoSelect on our website. I strongly suggest checking this out. I just wanted to highlight a few pieces of this where we show that cells grown in high-def B8 and sorted with this Wolf G2 sorter show the markers that we would expect for pluripotency, so specifically SSEA4 and TRAL160, and show high viability as well. And some images on the lower side are shown of single cells that adhered after sorting, after being grown in B8 after 24 hours, as well as healthy colony growth. So this is an image of a colony after 5 days grown in that medium. So if this is of interest to you and you want to look for gentle sorting methods of your cells, the Wolf G2 could be a solution for that. And we'd be happy to speak with you on that as well. Additionally, back in last summer, so 2022, we presented a few topics at ISSCR that have relevance in the cultivated meat space. On the upper left, we wanted to explore FGF2G3, so this thermostable form that we sell, specifically how its particular mutations confer its improved thermostability. So we worked on co-crystallizing this protein and solving an X-ray crystal structure of this. We show some highlights of this on that poster there, which is also on our site, to get a better idea of how these mutations could then be further improved upon to develop another version of a more effective form of this growth factor. Additionally, on the lower left, something that we also work on is medium optimization, and that's using DOE-driven methods. We used this on the B8 medium itself to look at multi-component improvements, so specifically looking at NRG1, insulin, and FGF2, and how those changes work in tandem to potentially improve medium performance. So strongly recommend taking a look. And last but not least, and certainly of relevance to the folks that are here on this call, looking at animal homologs of FGF2. So when we consider growing bovine or avian or piscine cells, looking at species specificity and looking at those changes across those species could give us information on if different species show improved performance or stability in the context of those cells or others. And we've seen this in the literature previously, where homologs of particular proteins can already out the gate show 2, 5, 10-fold improvements in performance and stability compared to a human form. And that's not always in the culture of human or other mammalian cells. We can see these trends also in other species, which begs the question, if we start producing other animal homologs of these growth factors, can that alone improve performance and reduce cost and bulk need of some of these proteins in the context of cultivated meat culture? So this work explored some homologs of FGF2 to begin. We looked at 11 different homologs across bovine, avian, and fish species, and we're now exploring those in preparation for ISSCR 2023 in the context of other animal cell types across the mammalian, avian, and fish species lines. And that's in collaboration with Tufts University, Virginia Tech, and some of our collaborators out here in San Diego. So we're excited to present on that work in June and hope to see you there in Boston if you'll be making it as well. Last but not least, and this is kind of an exciting development over the past year, we've had a lot of interest from folks in exploring medium and growth factor solutions for cultivated seafood. And obviously, there's a lot of work to do here on growth factor and medium development choice. One thing that we thought would help in this space is getting our own line of fish cells to explore how we can culture these and what it would be like to make a medium for optimization here. So our team went out on a fishing boat in the Pacific, and we brought a couple species on board, one of which was an amberjack yellowtail. We did a muscle biopsy right on the boat, which I can tell you that was certainly a fascinating experience for the team. Brought those samples back to the lab and started working with them, seeing what cell lines we could derive. And that work, after about 3 to 4 months of culture with Jerome here, who's on the call, resulted in one Pisces line from those biopsies that was tested in a variety of initial culture conditions. So that cell line has been taken to over 5 passages so far, and about 20 population doublings. And already its doubling rate has gone from 44 hours to about 24 hours or less at a culture temperature of 27 degrees. We can grow these cells in a serum-free and CO2 independent expansion medium. So we've already removed serum from the culture of these cells. And they're already showing adherence to TC-treated polystyrene, but they also show performance on a variety of ECM matrices. So it's been a lot of fun to work on this project in particular. It's taught us a lot about what the needs are for the cultivated seafood space. And we hope to provide more information on this medium formulation and our protocols coming up later this year and into next year to make this available to the team as a whole. But very exciting, promising efforts so far here. So for us, we're continuing to look at species-specific growth factors and seeing their performance in the cultivated meat space. This is in work with Tufts and Virginia Tech, and is also funded partially by the GFI grant from 2021 to 22. We're additionally working on high performance and serum-free formulations for cultivated meat. So currently our formulation B8 on the site is largely developed for human IPS, but it's shown traction for bovine cell culture, and we're exploring other areas as well and modifying that formulation and others for cultivated seafood and avian cell production. As well as expanding our capabilities with current and custom products. So getting into single cell culture and expanding these cells in suspension, that's certainly in particular going to be important when we consider bulk production of cultivated meat cells at a reasonable scale. And this is something we're seeing promise for already with high def B8 and looking into custom formulations as well for cultivated meat. So that was a bit of a kind of a whirlwind, but I wanted to briefly highlight our team as well. So Jerome and I are here on this call and happy to answer any questions related to what was presented here or any other questions you have about growth factors, media, and these cultivated cell lines. Fiona, Will, and Jose are part of the team as well, and we've recently brought on a stem cell scientist and 2 research technicians to expand our efforts in this space and others, and certainly have a wide range of expertise from our advisory board, both across the life science and pharma, as well as the cultivated meat space. So I want to thank you for the time today, and I think we certainly have time for questions, so I'm happy to dive into any specifics you're wondering about.
Thanks so much, Steve, for that very clear presentation. I'm glad to see all the work that you're doing. And yeah, as you mentioned, Steve is one of our GFI grantees, and we'll have others later in the year that present on this call. So I'm excited to start incorporating more of our grantee work into some of these seminars. Thanks so much, Steve, for the presentation. And I think people will know where to find you, and they can get in touch on some of the things that you described today. So thanks again to both of our presenters, and we will see you all here next month.
Thanks, everyone.